Hi everybody, so this is my introductory biology lecture discussing bacterial cells and their components. So we're going to talk about what bacterial cells are, what structures they have internally, what structures they have externally, and then talk briefly about bacterial cell motility. Before we talk specifically about bacterial cells, I want to talk a little bit about how we group cells and how we categorize them. So historically, up until just a couple of decades ago, we thought that there were two different kinds of cells, and we based these two groups on the cell's morphology, or in other words, the way that they're shaped. The two groups that we came up with were prokaryotes and eukaryotes. Prokaryotes were thought to called such because they did not have a membrane-bound nucleus. Instead, they have kind of this ball of goo called a nucleoid. Eukaryotes do have a membrane-bound, or quote, true nucleus. Another difference between them is that eukaryotic cells are much bigger and more complex than prokaryotic cells. We now know that that's a little bit erroneous. When we look at these from a phylogenetic standpoint, what I mean by that is by looking at their evolutionary history, it becomes clear that even though visually it looks like there are two different kinds of cells, in actuality there are three. Prokaryotic cells, in other words, these smaller cells that don't have a true nucleus, are actually two distinct domains of life: bacteria and archaea. Eukaryotes are still eukaryotes. So to talk about prokaryotic cells from a structural standpoint, their defining characteristics is that they have no membrane-bound nucleus, as we've said before. Principally, and in the rest of this lecture, and certainly in medical microbiology, we're going to be talking about bacterial cells, not archaeal cells. The size and shape of bacterial cells, and certainly their component, are highly variable, but most of them contain a plasma membrane, a single chromosome, so one big giant piece of DNA that encodes all the genes that they need. More and more, as additional genomes are sequenced, there do seem to be exceptions to this, but they do not have a large number of chromosomes at any rate, and they have only a single copy of any gene. They also have ribosomes, which synthesize proteins, and most of them, the overwhelming majority with a few small number of exceptions, have a stiff, rigid cell wall. When we talked about carbohydrates, hopefully we all remember what the component of that cell wall is, and if you forget, it's peptidoglycan, structural carbohydrate. I'm going to leave archaeal cell structure as saying that at this point it's not as well understood as bacterial cell structure, and what we do understand is rather diverse and not particularly well related to medical microbiology, so I'm going to leave it at that for the time being. So this would be a diagrammatic view of a bacterial cell, and here is a scanning electron microscopy image of a bacterial cell. And the diagram is pointing at certain cartoon structures that are overlaid with this TEM image. What you can see here is that this right here is this single chromosome. It looks like a big ball of DNA. It is in fact one piece if you spread it out. And that one piece is a closed circle. The other main thing you see in the cytoplasm are these little dots here. These are ribosomes. They're just floating around in all parts of the cell. This is very different from a eukaryotic cell where they're sequestered more or less in the endoplasmic reticulum. Bacterial cells do not have an endoplasmic reticulum. Another thing that you see here are something called plasmids. These are additional pieces of DNA. They're much smaller than the main chromosome, and it's important to note that they are not found in all bacterial cells by any stretch of the imagination, and they are not essential for life. A bacterial cell can gain or lose plasmids without it affecting its viability. The last two things that are being pointed at here that are common to all bacterial cells is right here is the plasma membrane. So this is a phospholipid bilayer with membrane proteins embedded in it. This is responsible for transport in and out of this cell. Around the plasma membrane you typically find a stiff cell wall. Again, there are wall-less bacteria, but there are not very many. On this diagram in the cartoon, we actually see two flagella as well. These are used in cell motility. We don't actually have them in the TEM image. Flagella are not present on all bacterial cells. They're certainly not essential for viability, but they are present on a fair number of them. Okay, so the genetic information in prokaryotic cells, again specifically bacterial cells, are contained in two different locations, and I just mentioned both of them on the previous slide. They're housed in what would be called a nucleoid, but that's not really a real thing. It's a term that's still used. Several people in modern bacteriology really don't find a lot of use for this term. The nucleoid basically refers to the big tangled up single chromosome, which can move all around the cell as it sees fit. Once again, there are exceptions to this, but the majority of bacterial cells have one single chromosome. There are some that have two that have been discovered. Those two chromosomes, again, are not matching one another. Every gene in a prokaryotic cell exists in a single copy. So we don't have any kind of matched pairing of alleles the way you would in a haploid eukaryotic cell. I'm sorry, a diploid eukaryotic cell. These chromosomes are closed circles, meaning there are no ends to these chromosomes. There are no telomeres on these chromosomes. They don't have a beginning, they don't have an end. They do have a place where they start replicating, called the origin of replication, but they do not have defined poles to them. Chromosomal DNA in a bacterial cell is not laid out like this. It's in a state called supercoiled DNA, and that basically means that this closed circle is twisted up, coiled up, and folded in on itself in several layers so that it can physically fit inside a bacterial cell. And also the degree and the shape of the supercoiling has a lot to do with which genes are expressed at any given time. What is also present and associated with chromosomal DNA are several proteins, and the proteins that you find in association with supercoiled DNA are usually involved in DNA replication, in gene transcription, things like that. Plasmids are another source, once again, of genetic information in a bacterial cell. By definition, a plasmid is extrachromosomal, meaning it's a piece of DNA that is not part of the chromosome. They're hanging around free in the cytoplasm. These guys are also principally circular. There are some exceptions to that. There are a few bacterial species I can think of that have linear chromosomes. The agent of Lyme disease, incidentally, would be one of them. And when these plasmids are circular, they are usually they are nearly always supercoiled as well. They're small. They're far smaller than the chromosome is. And in addition, as I said on the previous slide, these are non-essential genes. A lot of times they provide the bacteria with some kind of selective advantage. And if they don't, the bacteria are likely and inclined to get rid of the plasmids. If they're not providing them any advantage, they're not going to waste the energy replicating the DNA and transcribing it. I'm sorry, I forgot this was on here. Basically, the way that I tend to describe this in class, and I usually bring in a nice visual aid, is that if you had a giant rubber band laid out end to end, it takes up far more space than if you coil that up into a big rubber band ball. So think of supercoiled DNA as a rubber band ball and the actual chromosome as a big, giant, enormous rubber band. forgot this was on here too. I'm really sorry. You could tell that some of these slides I looked at beforehand and some of them I just started recording. This would be a just started recording lecture. One thing that I like to point out is that there's a bacterial species called Yersinia pseudotuberculosis. If you add 2 plasmids to it, and a few other genes, it is now known as Yersinia pestis. This organism causes gastrointestinal distress, so sometimes it's associated with infectious appendicitis, which certainly is not fun, but this doesn't cause crazy amounts of mortality, and in fact many of you probably have never heard of it. This organism here, whether or not you recognize the name, you probably have heard of because it's the causative agent of plague, bubonic, pneumonic, and septicemic versions alike. So these originally were called 2 different species because their disease manifestations were so very different. It was thought that they couldn't possibly be so closely related to each other. And in fact they are. Once we are able to look at them on a genetic level and perform genome sequencing, it's very clear that these 2 organisms are subspecies of one another, and a huge difference between them is that all pestis strains have 2 additional plasmids that pseudo-TB strains don't have. So plasmids not essential for viability, however they are in some cases associated with very meaningful behavioral changes in the organism. Okay, so supercoiled DNA is so dense that you can actually see it on high mag transmission electron microscopy. So this bit right here, these nice dark lines, this is actually supercoiled DNA. So this right here is not the double helix of DNA. Each of these bits right in here represent DNA molecules that are tightly coiled up, and so you can see this not only has secondary structure, it actually has tertiary structure as well. These little dots in here are bits of plasmid DNA. Okay, so I now want to just briefly describe some of the internal structures of prokaryotic cells. And one of the things that will immediately become clear is that internal structures in prokaryotes are extremely diverse. There are very few things that are indisputably in all bacterial cells, and certainly very few things that are in all prokaryotic cells. One of those things would be ribosomes. Every species of bacteria and of archaea have ribosomes. These are the structures that are responsible for actually synthesizing proteins, or you can think of them as protein factories. Ribosomes aren't just little structures that exist, they actually assemble as they're needed, and they assemble around pieces of messenger RNA. So when we have a piece of DNA that encodes a gene, and the cell wants to express that gene, it transcribes it and makes messenger RNA. That messenger RNA molecule actually sits right here and acts as a mortar between 2 pieces of a ribosome. When we talk about translation, we will describe this in much greater detail. But the other bit that's inside of pieces of RNA are not only something called ribosomal RNA, but you actually have, pointing to them right here on the screen, molecules called transfer RNAs that are responsible for actually, I would be doing air quotes, but you can't see me, that would be responsible for reading and decoding this mRNA. And then ribosomal RNA up here is responsible for adding amino acids to a growing polymer, which is ultimately going to become a folded up, mature functional protein. Okay, so ribosomes are in all bacterial cells. Chromosomal DNA is in all bacterial cells. And there's a membrane, at least, around all bacterial cells. And that is about it for the things they all have in common. Some things that can be found inside of prokaryotic cells are, for example, things like photosynthetic membranes. So these are unique to certain species, and as you could guess, they're unique to photosynthetic bacteria. I have this picture here, which is an image of a photosynthetic membrane, and in actual 201, I showed this picture again later in the semester because it looks very much like a chloroplast in a plant cell. And in fact, according to a theory called the serial endosymbiosis theory, a chloroplast basically is an internalized, now obligate part of a plant cell that was once a free-living photosynthetic bacterial cell. So that's kind of cool to think about. In addition to photosynthetic membranes, another thing that's present in some prokaryotic cells, but not all, is something called a cytoskeleton. So these are long, thin protein filaments, and they're very unique to certain species. I have an image here of a bacterium called Mycoplasma genitalium. So I alluded to the fact a few slides back that not all bacterial cells have cell walls. This organism is an example of one that doesn't. It only has a membrane around it. However, instead of having a wall over that membrane, it does have a skeleton underneath it to help give it some kind of structure and shape. That skeleton is also involved in helping this organism move. Some other things that are found in bacterial cells are what are now called bacterial organelles. The word organelles comes from, it just simply means little organs, and they're membrane-bound compartments that you find inside a cell. For a very long time, it was thought that the concept of organelles only existed in eukaryotic cells. These are fairly new discovery for prokaryotic cells. The reason why, and the defining feature here is the idea of having a membrane around something that's going to sequester it from the rest of the cell. The reasons why a cell would want to do this is that some chemical reactions are incompatible with one another. And so if you sequester a certain family of enzymes from their substrates by putting them inside of a membrane, that's going to keep your life cycle going a little better. A great example of this would be the idea that certain bacterial cells make enzymes called nucleases. Nucleases chop up nucleic acids. If you are a bacterial cell, you do not want your nucleases running around in your cytoplasm because they may chew up your own plasmids or chew up your own chromosomes, and you don't want that. That would be bad. So, among other ways of regulating this and managing this, one way you can do it is to sequester them inside of a membrane. Okay, so bacterial organelles are extremely diverse across species, and they're extremely diverse in function. They tend to be very, very specialized and evolved to work with the bacterial cell within its own particular ecological niche. So I have a table here, some of which describes some major bacterial organelles and some infamous ones, their function, how they're shaped, and what types of organisms have them. Everything with an asterisk, which is two things, are contained by all bacterial cells. Again, we have ribosomes and this term that I hate, nucleoid. But basically think of this as the tangle of chromosomal DNA. This little sunburst dealie here is meant to represent organelles that are present in some bacteria, not all by any stretch, but a great number. Those would be things like plasmids and flagella and carboxysomes. Everything that I, for whatever reason, marked with this little weird Spider-Man logo are present in highly specialized bacteria. So these would be things like magnetosomes. I have an image of these guys over here. These little black balls are magnetosomes. What they basically are a little pouch of iron filings with a membrane around them. And the point of these is that they're present in order to help these organisms move magnetotactically. And what I mean by that is that they follow along Earth's magnetic fields in order to move. It's pretty cool, actually. If you, and I'm sure if you search for this on YouTube, you could find a really cool video clip of it. if you put these guys on a hanging drop slide, so you have them in some nice liquid medium, and you focus a microscope with a camera on them, and you move a magnet across the slide, the cells all start chasing the magnet. And the reason they can do that is because they have these little organelles that tell them which way the magnet's going. It's pretty neat. But this is a type of organelle that's highly specialized to magnetotactic bacteria. Similarly, you have things like chlorosomes and thylakoid membranes that are unique to photosynthetic bacteria. I put this circle up here to show our Mycoplasma genitalium cell again, because it's got this funny looking knob on the end. This is actually called the attachment organelle. So this structure primarily exists to have this cell here attached to a host cell surface. But it's certainly not present in all bacteria by any stretch of the imagination. So that's why I have this little caveat here. There are many bacterial organelles that are not listed here and that do very specialized functions. Okay, so now I want to talk a little bit about external structures of prokaryotic cells. Basically, the three I'm going to talk about are flagella, pili, and capsules. So flagella, which is a plural word, if you're talking about just one of them, it's called a flagellum. These are these big, giant, long tails that you can see on the end or all over bacterial cells. And what these guys do is they're kind of like whips. They spin around and they move the cell in very powerful strokes and move it in one particular direction if they're only on one end of the cell. We will learn much more in medical microbiology about how these flagella are assembled and how they interact on the inside and the outside of the cell. In addition to flagella, we have a structure called pili. The singular, this is again a plural word, the singular is pilis if you're talking about one of them. So pili are much shorter than flagella, and they're much more fragile. Like flagella, pili are and can be involved in cell motility. So they wiggle around and they help propel a cell forward. Pili are also involved in cell adherence. So these guys, by their very tips, sometimes help anchor and stick this cell to a particular surface that they might be interested in sticking to. So those two guys are involved in motility and to a lesser extent adherence. The other external structure that I want to talk about are capsules. So this picture we've already seen before. These were Frederick Griffith's strep colonies, and the gooey, snotty S material on the outside, the smooth material, this is actually a capsule. And what it's made of is a big giant layer of complex glycolipids. So that's lipids with carbohydrates coating them, and they entirely surround the cell wall. Bacterial cells make capsules for several reasons, but the reason they're relevant and important in pathogenic microbiology is that they tend to help bacterial cells resist being engulfed by immune system cells. Can't say it much more complexly than that at this stage, but they're extremely important in pathogenic microbiology. We'll leave it at that. Okay, so I'm going to talk a little bit about bacterial cell walls now. Okay, so we talked about these to a small extent when we talked about structural carbohydrates. But bacterial cell walls, they're basically tough, they're fibrous, and they surround their plasma membrane. I want to make that very clear. Just because bacteria have a cell wall does not mean they do not have a membrane. Every cell has a membrane. And in the case of bacteria, the wall goes over the membrane. The membrane doesn't go over the wall. Another thing I want to make extremely clear, because it really bugs me when people, for whatever reason, don't remember this, is that animal cells and protozoal cells do not have cell walls. It is a major difference. between bacterial cells and animal cells and protozoal cells. And it is, in fact, as we discussed with structural carbohydrates, a very therapeutically useful difference between our types of cells. If animal cells had peptidoglycan cell walls, penicillin would kill us in addition to killing bacteria, and that would be bad. Okay, so bacterial cell walls come in a couple of different flavors, but the basic substance that they're made of, once again, is peptidoglycan, which is a structural carbohydrate, and the individual monomers of this big long structural carbohydrate are held together by beta glycosidic linkages. The two different flavors that bacterial cell walls come in are gram-positive and gram-negative. This refers to a specific type of diagnostic stain developed by a dude named Graham. We will learn how to do that in 315 lab, and you'll learn much more about how the chemistry of how this stain works. But the two different morphologies of these walls have retained the name of this diagnostic stain. The key difference between the two is that the peptidoglycan layer of a gram-positive cell wall is big and giant and thick. The peptidoglycan layer of a gram-negative cell wall is nice and thin and skinny. That's a huge difference. There are cell membranes underneath both of them. So the inside of the cell is down here in this case and down here in this case. The gram-negative cell wall has an additional membrane on top of it. So we have the cell membrane, the peptidoglycan layer here, and then an additional membrane over it. This entire thing, all three layers, are referred to as the gram-negative cell wall. Okay? Question I would pose if you all were in a lecture hall would be, does this matter to us? The fact that this is a big giant thick layer of peptidoglycan, this is a long skinny one, but it's got another membrane over it. Does it matter? And you can tell by the picture of Alexander Fleming who popped up in the corner, it does matter. From a therapeutic standpoint, it matters a great deal, because we need penicillin molecules to be able to chop this apart. We need them to be able to chop this apart. Is it easier to chop this or this? I actually have people discuss this and then tell me what they think the answer is. So you can think about that for a moment and pause the video if you like, but the actual answer is it's much easier for penicillin to chop this up than this, even though it's much, much, much thicker. And the reason for that is because in this case it has to get through a membrane in order to attack this, whereas here this is just out for the taking. The differences between these cell walls and the way things are transported in and out of these two different types of bacterial cells is actually extremely important therapeutically. It's extremely important pharmacologically, because we need to think about, if we're attacking something that is inside a bacterial cell, for example their ribosomes, we need to think about how to get that compound past this wall and through this membrane, or through a membrane, through a wall, and through another membrane. So it matters quite a great deal, the structure and the chemistry of these bacterial cell walls. The last thing I want to describe is bacterial cell movement. So all of these links here will take you to different video clips of specific cells moving in the way that are being described. I'm sorry, I've been doing this all day and I am losing my voice. The first type of motility is movement by flagella. In this case we have a cell with one flagellum, and it's using it as a big long whip. So when cells move using flagella, they tend to move in great big bursts, and then they whip the flagellum again and they move in another burst. So flagellar motion is a little bit jerkier than some of these other types of movement. And you can actually see that if you watch these video links here. This is fairly unusual, this scenario here. What's more common is that you have a bacterial cell with a whole bunch of flagella all over it. And a lot of times they're coordinated and they all whip at the same time, but other times they're just kind of going nuts and they're whipping as they see fit, as it were. So this type of cell can sometimes have very, very jerky motion as it moves forward. If it's deciding that it wants to move toward or away from something in particular, it will coordinate these flagella much better and get them to move it with a very nice speed in one particular direction. Okay, so pili are a little different. These are shorter than flagella but longer than cilia. And these are filamentous, they're fragile, they're also kind of like whips. They tend to be all over the cell's surface, much shorter than flagella are, and their motion is not nearly as strong. So when you have movement by pili, you tend to have much smoother motion. Same goes for cilia. So cilia are very, very, very short, and they're almost like little hairs that come off the surface of the cell. And this is actually a eukaryotic microbe now as I look at it, but these guys just kind of wiggle, and so these move a lot slower, but the motion is much more fluid, even more fluid than pili. Another type of bacterial motility that we want to know about is gliding motility. So this is done by protein propulsion, and it's done, the proteins that mediate this are not just one family. It's not just a common family of proteins such as those that make up flagella or pili. Gliding motility seems to have independently evolved several different times in several different types of bacteria. But the structures that mediate this are partially internal, partially external. And if you click this video, you'll see that this is the smoothest motion you can see. Very, very, very uniform. The last type of motility that we want to talk about is motility involving endoflagella. So this is kind of an interesting situation, and this is typically seen in types of bacteria called spirochetes, which are shaped like this. They're shaped like corkscrews. Endoflagella are flagella. They're structurally just like this. They're just as long, they're just as strong, but they're not sticking out of the cell. They're actually embedded under the membrane. And they tend to cluster on one side of the coil and one side of the spiral or the other. And so when these guys all whip at the same time, they actually get the entire cell to move in a corkscrew motion. And so I have a video link right here that's looking at a I believe it's the Lyme disease spirochete, but I'm not positive on that one. Hopefully it says in the caption. I believe that is the last slide in this lecture, so... Oh no, I lied. I have the summary slide. So we have, what we want to take away from this for bacterial cells is that we have essential subcellular components that are present in every bacterial cell, and those are circular supercoiled chromosome in a, quote, nucleoid, but again, I hate that word. We have ribosomes, and we have a cell membrane. By and large, we usually have a cell wall also, but not always. So possible components you can have inside the cell that are reasonably common are things like plasmids. Less common, we have specific bacterial organelles and the cytoskeleton. Another point to take away from this is that cell walls in bacteria are extremely important from a clinical standpoint, And they're also extremely important from an evolutionary standpoint. They're very evolutionarily telling. All gram-positive bacteria are more closely related to each other on a genetic level than they are to any single gram-negative bacteria or bacterial species. So this phenotype of having a gram-positive or a gram-negative cell wall actually correlates nearly perfectly with their evolutionary origins. If you have no cell wall or a weird cell wall, that caveat doesn't apply. Just as an FYI. The last point is about motility. Some species are motile, some species are not. Of the species that are motile, there are several different mechanisms that they use to move around. And that's all we have to say. I really should practice these more before I give them. Okay, I'll do better next time. Thank you.